recording. Okay, so I think it's recording now. So good morning. This is Wednesday, the 2nd of September. Really excited to talk to Gabor Erdosi, who is a background of molecular biology and is going to talk to us today about lipid metabolism. So my name is BarlowCollins. I'm a GP and have been for the last 25 years. And I've been involved in transformational work within the NHS, usually focusing on processes. But over the last few years, what I found out was we might improve the process, but we don't fix the root cause. So we can get our diabetic clinics running very efficiently, but we're not stopping the tidal wave of diabetics coming to us. And in our network, we've noted that we have pre-diabetics in the region of about 1,100 and type 2s in the region of 1,200, but we're converting them at the rate of 12% per year. And I know that is utterly unsustainable. So we need to have a new way at which we look at root cause. And Gabor today is going to talk to us about, particularly about lipid metabolism. And I know in my course of learning within the NHS and within my medical school, my knowledge of lipid metabolism is extremely low. And I think most of my colleagues would probably quietly admit to that as well. So as a result, we revert strictly to guidelines without thinking about why we're doing what we're doing. So Gabor, can I get you to introduce yourself and we can start our chat?
Yes, thanks, Donald. As you mentioned, I'm a molecular biologist by training, but actually I stopped believing that training is extremely important for what you are doing. I think that research is the most important thing for what you would like to understand. And in fact, I spent more than a decade working with surgeons initially in the operating room with different surgical devices and stopping bleeding and these kind of things. And then I moved to industrial biotechnology, and I spent almost 12 years supporting and working in the grain processing industries, such as high fructose corn syrup. It's very interesting and everybody loves to talk about this, and maltodextrins, different modified starches, alcohol producing, fermentation, and also in brewing, so a little bit of baking as well. So I was kind of a industrial expert supporting these companies how to produce these foodstuffs or food components at an ever decreasing cost. So that was the main reason we sold them enzymes, but supported them with their processes, optimizing the use of biotech in their processes. So and it's quite obvious from this that these had exactly nothing to do with my current interests. So I arrived to this very similarly to you, that I was overweight. My mother was worried that what was going to happen with her grandkids, because my health was deteriorating, and yeah, I started a weight loss journey at the end of 2013, so it's almost 7 years now. I lost something like 32 kilos. It's something like 70 pounds or 65, 70 pounds in half a year, or a little bit over 5 months, very quickly. I had no intention to lose that much. I had no intention to lose it that quickly, but it just came off. And eventually, as a guy involved in complex problem solving in the industry, and having a background in biochemistry, molecular biology, I developed this unsatisfiable curiosity of how on earth this worked so well. I mean, I couldn't believe that I was able to eat anything or any amount I wanted, and I could still lose weight. It sounded so unnatural. You know, we heard that we kept hearing eat less and this will resolve and move more, and then it's easy. And I was feeling that I was eating more. Most likely I wasn't, of course, but I had the feeling that I was playing around, grabbing a Brazil nut and putting butter on top of the Brazil nut and eating a handful, and was curiously doing this experiment for 2 weeks and observing if my weight starts going up. But no, I kept losing. So I was just astonished, and I wanted to understand that that obviously there was something wrong with what we were being told. It's not really a basic equation between what you eat and how much you exercise. Of course, the underlying physics always applies. There is no doubt that there is an energy balance and imbalance going on. But the drivers, the underlying drivers, are not what we typically believe, I think.
And how was your hunger during that time?
I was not hungry. I was absolutely not hungry. I ate, typically ate meat, animal stuff and vegetables. And later on, I added fruits as desserts. But I was absolutely not hungry. And one of my online friends named this diet as the meds diet, like with the medications, the short meds. It's meat, eggs, dairy, and what was the S? I don't know. Seafood. Seafood. So it's a meds diet. It's like coming from the medical pharmaceutical industry. Just it contains meat, eggs, dairy, and yeah. And then you can add the plants. If high carb plants, you can add them as desserts. If low carb plants, you can add wherever you want.
Wonderful, wonderful. So, can you tell us something about the fat in our body? So, as in the fat cells, because we've got different types of fat cells, and what's their purpose? And because I know there are some animals that don't have fat, and us humans have an increasing amount of fat, and they must have some function.
Most of the animals do have fat in their bodies, but some of the more ancient type of animals don't have dedicated tissues storing fat. So, for example, insects have a so-called fat body, but it's more like a liver than an adipose tissue. So it's kind of a combination of liver and some fat storage organ together. It's a metabolic regulator, but it combines the functions of a liver and a adipose tissue, a fat storage organ. Humans indeed have considerably higher amounts of body fat as normal, comparing to other primates or higher apes. Comparing to chimpanzee, I think we have at least double as the normal. A adult chimpanzee male has something like 4-6% body fat, and this 10-14% is more or less the normal for an adult human male, and females have even higher levels of body fat as still as normal. They can go up to 20, even 25%, and operate perfectly without any metabolic disturbance. So this shows that we have evolved to store and carry around more fat normally. So this shouldn't cause issues. But something obviously happens, and it has accelerated in a big way over the last couple of decades. So there is a connection between becoming more obese and becoming metabolically deranged, so to say. And the connection is not so straightforward as many would believe. You know, there is this body mass index, BMI, which is often cited, it only approximates your body fat by dividing your height by your weight, and then
Surface
taking
area.
the square area, surface area, yes, and it approximates your body fat content, but it also excludes the differences in lean mass, muscularity, and these kind of things. So it's really just an approximation, but it still correlates quite strongly with diabetes, pre-diabetes, and insulin resistance, for example. So there is an apparent correct connection between the two, body fatness and metabolic problems. However, not all fat depots were created equal. That's also obvious. We have the so-called subcutaneous fat under our skins. Basically, that's a technical term for fat under, or anything under the skin, subcutaneous. And we have this visceral type of fat, which is also called intra-abdominal. Abdominal is within your belly. It's belly fat, otherwise, in a more simple term. So the difference is that these fat depots have different physiological roles, different metabolic profiles, and basically react differently to different stimuli. So what happens when you stuff yourself is you create more inflammation in your visceral fat, because that's close to your digestive system, and in fact it supports your digestive system with the immunologic um, functions. While the role of the subcutaneous, the big mass of fat under your skin is more a storage one and less a metabolic regulation one. It's this happens on a spectrum, so it's not like black and white that this type of fat is only metabolic and immune supporting, and the other type is only um, storage fat. But this is a spectrum, and there are distinct differences between the two. So this is how lipids are stored and then released. Your adipose tissue, the fat cells in your body, take up the lipids coming in with the diet, the fat basically, and then they also release when needed. When you don't eat for a couple of hours, they increase the release and reduce the uptake of lipids. That's the dynamics. That's the basic dynamics. And then we have two more organs which are deeply involved in lipid metabolism. One is, of course, your small intestines, the small bowels, when the lipid absorption occurs from food. So you ingest the lipids in your food. When you eat bread and butter, for example, butter, of course, is almost pure lipids, and these lipids are broken down in the small intestines by enzymes, and then the liberated fatty acids are taken up by the small intestines, and then they are packaged into so-called lipoprotein molecules. These are small balloons, balls, which contain polar, water-soluble compounds on the outside, because fat is not water-soluble. And then the insoluble, the lipid parts, go to the inside. And then this is how they are carried around in the blood and also in the lymphatic circulation. In fact, there is a distinct difference between different chain lengths of fatty acids, how they are handled by the body. Short chain fatty acids, acetate, acetic acid is in fact a short chain fatty acid. Butyrate, propionate, these are short chain fatty acids fermented by gut bacteria. And there are medium chain fatty acids, medium meaning between short and long, of course. And these are taken up into the blood, which reaches the liver immediately and directly. That's because these compounds cannot be stored. So short and medium chain fatty acids has to be burned or processed to something else. Otherwise, they cannot be stored in our body. So that's a major distinction. And then we are left with long-chain fatty acids, which can be stored and which are the major storage form of lipids. These are packaged into these lipoproteins we mentioned, and then released into the lymphatic circulation, not into the blood. The lymphatic vessels are part of our immune system, and there is a good reason why they are, because the bacteria in our gut also release these lipid-like compounds. And these are taken up together with the long-chain fatty acids. And you don't want them to reach the liver directly because they can be highly inflammatory, and you don't want to create a inflamed liver. So you send these particles together with the bacterial compounds through your immune filters, so to say. This is exactly what visceral fat, the intra-abdominal fat depots do. One is attached to your intestines and called the mesentery. This is a long fatty organ. Now it's considered a separate organ. And it contains a lot of lymph nodes. Lymph nodes are part of the immune system. They contain immune cells, and they are structured in a way that these lymphatic vessels deliver the stuff collected from different tissues, and these lymph nodes filter out all the antigens which are in the lymphatic fluid. So basically, this is what happens with lipids taken up by the intestines. They are filtered by these immune organs, your mesentery and mesenteric lymph nodes, and also there are lymph nodes in the pancreas, and some attached to the stomach. So this is the start of the uptake of lipids. Then these lipoprotein particles reach the venous circulation, eventually just before entering the heart. Then the heart pumps out the arterial blood, and these huge lipid-containing particles reach all those tissues which require lipid either for energy or for storage. I think after a meal, the vast majority goes to storage, because you don't need that much immediately. Of course, if you have 3 main meals in a day, you don't need the energy immediately after those meals, but you need them gradually over the 24 hours in a day. So that's why the dynamic is that you eat the lipids, and actually you release the lipids from the previous meal, mainly from the intestines. It's a little bit more complicated than what happens to glucose, because most of the lipids released after a meal come from the previous meal, but don't let you confuse. Don't let you be confused by this. It's a major, it's a minor thing for understanding the basics. It has some important implications later on, but basically you have to store the fat, and you can store, if not any amount, but you can store a huge amount of fat from a meal. So that's a major difference compared to carbohydrates. You have limited storage capacity for carbohydrates, especially if your stores are not empty. So if your meal occurred just a couple of hours after the previous meal, your liver stores carbohydrates as glycogen. Also, your muscles store carbohydrates as glycogen, and if you don't empty your stores, it has to be burned. So there is no high storage capacity. For lipids, you have almost endless storage capacity. So it goes to your adipose tissues, fat cells. Again, there is a need to break down the fats, and then on the other side of the cell membrane, it will be assembled again to the storage form. And the storage form is what we call triglycerides, abbreviated to TG or TAG, because it can be triacylglycerol. That's the more scientific name for the compound. So it's TG triglycerides. That's the most common That's what you store.
Yeah, and that's the same compound we measure in the blood. So when we get the
Yes.
lab result back, yeah.
Yes, actually, but the funny thing is that we don't measure triglycerides. Triglycerides, the name comes from, and more precisely, the triacyl glycerol comes from, you have 3 fatty acids, long chain fats, and you have one glycerol molecule which looks a little bit like half a glucose. And then you esterify, you attach the 3 fatty acid molecules. Yeah, let's show 3. So to these hydroxyl ends, and then you have this triacyl glycerol, and actually it's immediately proportional. You have 3 fatty acids, and then you have 1 glycerol. And how in fact this triglycerides is measured is measuring glycerol.
Okay.
So you digest in the little test. You digest. This is a test strip for triglycerides, for lipids. It has to be digested. So you release from this triglyceride molecule, you release the 3 fatty acids, and then you can most easily measure the glycerol. And then you know that how much triglycerides you had, because every single glycerol molecule was a triglyceride molecule before. So it's a one-on-one calculation. It's not, there is nothing weird about it. But you have to digest the triglyceride to be able to measure, actually.
So the pathway you talked about, where going through the mesentery and then going around the blood vessel system. So is that why we pack our pancreas and mesenteric visceral area first?
Yeah, you mean why people tend to develop this beer belly with high intra-abdominal visceral fat and also pancreatic fat? Yes, I believe actually that's the reason. When the stimulation from the intestines, the inflow of bacterial debris increases, then the demand for immune clearance, for filtering, also increases. And then your lymph nodes become bigger, and also the supporting fat cells around the lymph nodes become bigger and more numerous. So basically you have a filter organ which expands with demand. This is how I see it, at least.
Yeah.
So this is what happens.
And you talk about bacterial products getting filtered in there. So if we have an abnormal gut microbiome because of poor diet, presumably this puts extra pressure.
Yeah, it's difficult to actually disentangle which comes first. It's kind of a chicken and egg problem, because when you have an inflammation in the body, the inflammation also can disturb the immune function in the intestines. Obviously, you have an inflammation, different immune cells become activated, and the immune balance is tilted. This can also cause problems with the bacterial composition, because there is an intricate balance or interaction between the host immune system and the bacterial communities within, or mostly attached to the intestinal wall. And I have had problems disentangling what actually occurs first. But it can become a self-perpetuating cycle, for sure. So it might not be that important whether the food causes altered inflammatory responses in the intestines, and that causes the problem with the bacterial communities, or the food directly causes a change in bacterial communities which stimulate a change in the immune response. I think both are plausible. And it's perhaps, as I said, a self-perpetuating kind of a vicious cycle after a while. But how it starts, I'm not 100% sure yet.
Okay, and can you tell us something about the role of gut hormones and fat cells? So there's these hormones in the upper gut, GIP, and we got insulin.
Yes. you actually, most people are not aware that the hormone insulin, which is kind of a anti-catabolic hormone, it prevents the breakdown of fat and stimulates a little bit also the storage of fat, and also stimulates the storage of glucose. It stimulates storage and puts a brake on breakdown. So that's the main role of insulin in the body. And a lot of people are not aware that insulin is mainly not directly stimulated by food components. I think that the traditional view was, and in some folks still remains, that you eat, for example, glucose or protein amino acids, and these stimulate insulin in the pancreas, in the beta cells, which release insulin directly. But actually, this process has been substantially refined over the last couple of decades. What really happens is that, yes, your blood glucose also has an impact on insulin secretion, but the majority of stimulation comes from your intestines, by intestinal hormones, which are called incretins. And these incretins, what they do, and there are other compounds on top of incretins. For example, fatty acids can also have an influence on how glucose-stimulated insulin release works. But let's stick to the intestinal part. It's up to 2/3 or up to 70% of released insulin in response to, for example, glucose is not directly mediated, but by these so-called incretin hormones. So what happens is that glucose is taken up by the so-called epithelial cells in the intestines. These are make up the intestinal wall. These cells, it's basically a single layer of cells which take care of producing the mucus in the intestines, but they also take up these specialized, so they take up food compounds, and some cells specialize to release hormones. These are called enteroendocrine cells, and they can take up, for example, glucose or amino acids the same way as regular cells, regular intestinal wall cells. But what they do, that in these cells, the uptake is coupled to hormone release on the other side of the cell. So from the lumen, from the internal of the intestines, they take up, for example, glucose, and this glucose stimulates hormone response, and the hormone is released to the blood on the other side of the cell, the enteroendocrine cell. So what they do, that these hormones reach the pancreas, or they stimulate nerves, and this circuit feeds back to the pancreas, and they stimulate insulin release coupled to glucose. So you need both glucose and these hormones, but what actually happens, these hormones are responsible for the majority of insulin stimulation. Without these hormones, if you inject the same amount of glucose intravenously, you only get 1/3 of the insulin response. So that's what they do. And it turned out that we have different kinds of these hormone secreting, incretin secreting cells in the intestines, and they are not evenly distributed. And it has major implications. For anybody interested, look up the Food News lecture I gave in Prague 2.5 years ago. I summarized that. Then Dr. Michael Eads also presented about it in the US in a low-carb conference a year later. Perhaps it's in more understandable English than mine, so you can also benefit from you viewing that, watching that. What happens is that these cells are not evenly distributed. So some cells are more populous, more higher in number in the upper intestines, and other types in the lower. And what I realized a couple of years ago, that when you eat foods that humans are evolutionarily adapted to eat. These hormones are stimulated in a balance. They go up and have these similar levels, and basically what happens is they have multiple functions in the body beyond stimulating pancreatic hormones. They are involved in lipid storage. They are involved in inflammatory and anti-inflammatory processes. So what you really would like to see is that they are remain in balance. Unfortunately, what we do with our food by processing the hell out of it is that we create a huge imbalance, because the food is basically pre-digested in industrial processes, and you don't have much to do with this food. You can very quickly break down the remaining chemical bonds between the molecules, and you can absorb all this food in the upper small intestines. You only use a small part of your digestive system to deal with these ultra-processed foods. While in contrast, if you eat meat and vegetables, for example, it's protein, fat, and roughage, basically tough, fibrous food. It takes a lot more time to break it down, and all the nutrients included are absorbed in a more balanced and more gradual way. A lot more of this food stuff reaches your lower intestines. That's basically the difference, anatomically and functional physiologically, that happens. And as it turns out, and I realized a couple of years ago, it has major implications for your health, for your metabolism, for fat storage, for everything, for immune function, immune stimulation. And no wonder, I mean, our bodies have evolved over millions of years, even since demerging from the common ancestor with apes 2.6 million years ago. We have evolved to consume these difficult to digest foods, and not this pre-digested industrial stuff. I mean, it's like you don't need teeth, you don't need a digestive system. You are an absorption machine. If you consume industrially ultra-processed foods, you are an absorption machine. And go ahead.
And what's interesting with that is there anywhere in nature where you get sort of fat and sugar combined?
Not really.
Yeah.
I mean, I cannot really tell you one single foodstuff which is high in both. It can occur in both. I mean, you can eat, for example, peanuts. It contains 20% carbs and 40 or 50% or even higher amounts of. So it's relatively high in both, but it's still not similar to what you get in a donut. A donut is 50% fat, 45% quick digesting carbohydrates, and 5% protein or something like that.
And what's interesting when you mentioned donuts, at the start of the COVID crisis, Krispy Kreme is a big donut maker over here. And they donated millions of donuts to the NHS hospitals to
good luck.
help them in this health crisis.
Yeah, that's a different problem, because again, I can just refer you back to my talk in Prague, that this creates huge glucose fluctuations in the body, and not only upwards but also below baseline, and that's really bad. I mean, we have started recognizing over the last couple of years that glucose variability, so these up spikes and dips, are even more detrimental than continuously high blood glucose levels. So this is a big problem, and it has a huge impact on immune function. If you are in a hospital dealing with an infectious disease, or basically any disease, your immune system is involved. If nothing else, in healing, then you shouldn't bombardier them with these glucose spikes and dips. That's a really bad idea.
So what you just talked about all this stuff that happens in the upper gut. So presumably this is why bariatric surgery is so effective.
This is one reason, at least I suspect so, that this debate is still ongoing. There was a study recently which showed that all the benefits, all the very quick benefits, because what you see is a much quicker resolution of diabetes and insulin resistance in a matter of days. If you measure things in 5 days or 1 week, you have profound improvements in insulin sensitivity, and your glucose tolerance, for example, improves in a profound way. But there was a study very recently which showed, or I would say tried to show, that the vast majority of these benefits comes not from the operation itself, but the fasting and weight loss preceding the operation. But I think it's not true. Still not true. I'm not a guy who pushes for weight loss surgery. Or now it's also called metabolic surgery, because normal weight people can undergo this operation to get rid of try to get rid of their diabetes. And it's fascinating to learn from what happens after this surgery. But I don't think that this should be the first option offered to people struggling with weight or struggling with diabetes. But as I said, it's absolutely fascinating to observe and try to understand what happens, what is behind the very quick changes in health markers which occurs after the surgery, and that occurs before any major weight loss happens. So it's not closely tied to the longer term weight loss benefits of the surgery. And yes, one of the suspected mechanisms coming from weight loss surgery is that they do this, at least in some types of procedures, they do this bypass. They bypass the first part of the small intestines. They cut off the outlet of the stomach, and then make anastomosis. They reattach it further down to a lower part of the small intestine. So what they do is just a shift of nutrient delivery from the upper to the lower small intestines. And it's the most easily observable is in a procedure which called Biliopancreatic Diversion, which means basically that they don't do the butchering of the stomach, which all other procedures include. They don't do the butchering of the stomach. They just do this bypassing of the upper smaller intestines. Actually, there are some developmental experimental procedures when they enter a sausage peel. I call it a sausage peel. It's basically a nylon tube, flexible nylon tube, which they enter into the outlet of the stomach. So the proxima, the first part of the small intestine, it's something like 40, 60 centimeters long. I don't really know, something like 15, 20 inches if some Americans watch this. And it also has the profound impact on metabolism. Both procedures, this experimental one with the sausage peel, and the other one, the operation when they just bypass the first part, the BPD, the biliopancreatic diversion. And in several studies, they don't do the pre-operation weight loss regimen. So and it does occur without the pre-operation weight loss. So I think these authors suggesting that all the benefits of this bariatric surgery procedures comes from the pre-operation weight loss is not true, because most of these procedures use the weight loss. It's very difficult to separate the effects. But some of these procedures don't do, and some of these procedures don't even do the butchering of the stomach. So it's not, it doesn't happen because you cannot eat a lot because your stomach, 2/3 of your stomach were cut out. was cut out. So
yeah. Or of course, the other option is doing what you and I have done, which is to remove the ultra-processed foods, reduce your sugar intake, and you get similar outcomes.
Change the absorption profile,
Yeah.
and then you are back to normal. Or you can even do a little bit of reverse imbalance, which
Yeah.
the bariatric surgery does. It
Yeah.
creates a reverse imbalance. So it suddenly creates a healing type of, not disease making, but a more healing type of imbalance,
Yeah.
which also resolves by time. After 1 or 2 years, these changes in food delivery and the distribution of these hormone secreting cells actually adapt in the intestine. So the primary trigger caused by the bariatric surgery procedure is gradually being removed. You won't stay forever in this, and a lot of people actually gain weight back after bariatric procedures. 1 or 2 years later, they start gaining weight along with these changes.
Yeah. So one of the things I learned from your recent, from that lecture in Prague, was about your mixed meal challenge to the system. So as we slowly develop metabolic syndrome, if we challenge the system with these mixed meals, there are things we can measure to demonstrate what's happening.
Normally, when you undergo a tolerance test in a lab, prescribed by a doctor, such as you, it's an oral glucose tolerance test, and then you can get out different glucose curves, different glucose profiles. Even if your fasting glucose is normal, your reaction to a lot of glucose can be abnormal, and then you can tell a patient that you are pre-diabetic or diabetic based on the, for example, 1 hour glucose level after this tolerance test. But most people don't consume pure glucose at home, so it's not a standardized way of telling anything. And also, your metabolism is a bit more complex than consuming glucose. You normally eat mixed meals. You eat a piece of meat with potatoes and vegetables, and have a dessert afterwards. So it's a whole mix of protein, fats, some fibers, and glucose, carbohydrates. So it's and as soon as you eat a mixed meal, regular mixed meal, these profiles in glucose, postprandial glucose, disappear. So you are unable to tell if somebody is glucose tolerant or not if they consume a mixed meal. However, if you start measuring other parameters and you really increase the size of the meal and keep the meal composition similar to resemble both a glucose tolerance test and a lipid tolerance test, while keeping protein at the normal levels, you can really stress the system, stress metabolism, and see if metabolic flexibility is there or not. And what you see with metabolic flexibility is basically insulin sensitivity or insulin resistance, the opposite. So where this guy falls on the spectrum of insulin sensitivity. He can remove the lipids from the blood and the glucose from the blood readily. It doesn't go up very high. I mean, neither of these go up very high, and they go down as expected after a few hours, and they don't linger high after 4 or 5 hours. So you are able to do a different type of tolerance test, which is more telling, which adds additional information compared to a glucose tolerance test.
What would you test in that?
Then we are back to lipid metabolism, and why this talk is focused on lipid metabolism, because it turns out when you start looking at different parameters in the blood, that what first fails is not glucose uptake or glucose release in the body. But what first fails is lipid uptake in the body. And lipid uptake is impaired because your fat cells start becoming less insulin sensitive. So that's kind of a first event in insulin resistance in the long course of developing higher and higher insulin resistance. What occurs? You are first only categorized as insulin resistance, and then having metabolic syndrome, prediabetes, and diabetes. This is a long course of the same disease, so it can take decades to develop diabetes. And then looking at fasting glucose or prediabetes when your fasting glucose is still normal, but your meal tolerance glucose can be abnormally high. It turns out that lipid metabolism deranges much earlier. And it's also likely that glucose, all the derangements in glucose metabolism is the outcome, the result of the derangement in lipid metabolism. So actually following lipid markers, especially after a high lipid, high glucose tolerance test, makes a lot of sense, because you can, this way you could catch abnormalities a lot earlier than using a glucose tolerance test only.
And so in specific, so I'm sitting in my GP surgery, so I'm hearing what you're saying, but what am I actually going to measure? So
ideally, you would be measuring free fatty acids in the portal circulation, so the blood which goes from your small intestines to the liver.
yeah.
So that would be the best marker. But obviously, you cannot use a 30-centimeter needle and try to somehow puncture this huge vein going to the liver, because that's a hugely invasive procedure, first of all. And secondly, measuring free fatty acids is not an easy one, even in a highly accredited lab, not to mention a home setting or a small GP office. So we need to use proxies, and the next best lipid proxy is the mentioned triglycerides. So triglycerides, there are two types of triglycerides in your blood. One is released from the small intestines and traveling around in particles called chylomicrons. These are the biggest, the largest particles, because they carry a lot of food lipids. And the other one is so-called VLDL. A lot of people heard about LDL, so-called bad cholesterol. And this VLDL is very large. LDL is released by the liver. So it's kind of part of a lipid cycling. The liver receives free fatty acids from fat storage, and then what it doesn't need, it assembles into these VLDL particles and sends it back to different tissues, either for storage or for the heart, for operation, and these processes. But these are the two main kinds of triglycerides in the blood. When one eats a meal, obviously the chylomicron type of triglycerides, the food-derived triglycerides, dominate. That's obvious. When fasting, for example, the next morning after a early dinner, 12-hour fasting, then the vast majority of triglycerides in the blood are contained in these VLDLs released by the liver. So that's the difference between measuring fasting triglycerides and postprandial, after a meal triglycerides. After a meal, the vast majority is in chylomicrons, meal type of triglycerides. And these are mainly taken up by adipose tissues for lipid storage, because you need to distribute your energy energy demand is distributed over the 24 hours of a day. But your meals, for example, if you have 3 major meals in a day, that is not evenly distributed, obviously, throughout the day. So you need to store and later release. That's what adipose tissues do.
Okay.
And if adipose tissues fail to properly uptake enough lipids from these chylomicrons, then what you see is a different lipid curve after the meal. So that's where you can spot a relatively early derangement in metabolic health.
And I guess this is where this home measuring device will come in, because you know, in my GP surgery, I don't really want people sitting there for 6 to 8 hours and me doing
Yes.
bloods repeatedly over.
Yeah. I mean, you can do a fasting, of course, if the patient arrives fasting, you can do a quick check. It requires, actually, these devices, I have one at hand. These devices can do both glucose and lipid measurements.
Okay.
And it requires the same amount of blood what you take for blood glucose. It's 5 microliters. It's an extremely small amount of blood. And you can get out the triglycerides, the HDL, the good cholesterol, the total cholesterol numbers, and as well as the blood glucose. And you can immediately create at least 3 different indices of already from the fasting measurement. But if you would like to, you suspect, for example, you see that your patient has a nice little beer belly.
Yeah.
You suspect that he's in early stages of seeing issues dealing with food. Then and all his fasting measurements are normal.
Yeah.
You could still do this tolerance test and tell the patient, okay, eat this meal, and we take the 0-point measurement here, and you can do the, I don't know, 1 hour, 3 hour, and 5 hour finger prick at home.
Yeah.
It's a very simple measurement. Here we do the 0-point. You see how to do it. It's extremely easy. And please do the 1 hour, 3 hour, and 5 hour, and just jot down the results. And then you come back with the results, and we analyze your meal tolerance, basically.
And then we can easily plot that on a graph, so I can easily see if it's an abnormal response or a normal response.
Yes.
Yeah.
It can also be automated. I work with a project called metabolic.
Yeah.
We try to do exactly that, using some of these easy devices, but also your phone camera to do some of the measurements. So you use a blood test, and after that you need to take a picture of the small test strip. And based on that, the algorithm in the cloud will be able to analyze the density of the color on the strip and come back with a very good approximation of your, for example, insulin or C-peptide or C-reactive protein, or even interleukin 6, these inflammatory molecules measurements after a meal, for example.
So it's extraordinary. These phones can effectively do chromatography.
Well, yes, basically yes. Although it's not the phone that does the trick; it's the computation in the cloud that takes all the information and then analyzes, spits out the absolute numbers, and then plots the numbers, plots the measurements on a nice graph.
So what's interesting for me is trying to get to root cause. So if I have a population covering my network of 40,000 patients, and then I can pick out a group who have an increasing waist circumference or an increasing weight with maybe slightly rising blood pressure, that might be an interesting group to do these things on.
Exactly. Although, yeah, it depends on when one would like to, if somebody is interested in his own health, of course, and doing these measurements at home, in a home setting, they would like to know where they are on the spectrum. Even if they have a flat belly, they can have some level of issues. It's not a perfect correlation, but in the general practice, I think, it makes sense if you see somebody with a little bit of protruding belly, a little bit of double chin, and these kind of things are good signals of metabolic issues. And then, even though their fasting measures can be in the perfect range, but their postprandial handling of nutrients, this is where the body fails first. So that you would like to point out as early as possible.
And I'm seeing increasing numbers of people with, skin reactions. This thing called acanthosis nigricans. You can see that in the street,
That's also a typical
skin,
insulin resistance symptom.
Skin tags.
Yeah.
So
there are many. There are
yeah.
many of these symptoms which people don't even realize that represents a problem in their bellies.
And the challenge, so within our population, often is that the biggest group will be in deprived populations, so people with low socioeconomic backgrounds, low education levels, so they might not be aware that their health is deteriorating because, quite frankly, they look like a lot of their neighbors. So I feel comfortable that I'm just like everyone else.
Yeah, and here in Europe we have general access to healthcare at least. Yeah. So they can have access to the same measurements that anybody else, but for example in the US it's not like that. So yeah. it matters a lot actually if you can reduce the cost. It also matters here. I mean, NHS in your country will be extremely happy if you can cut some costs into half. I'm sure they don't resist. We tend to measure all these markers based on historical developments and not really on current science. Some of these could be left out, others included instead, different time points being used, different indices used, and then actually you can even save on how much you measure, by what and when you measure. You can get out the same quality of information by using lower quantity of and cheaper measurements.
Yeah, I mean, this has been fascinating, and I think for me, I spent the first 20 years of my career looking at slightly rising triglycerides, slightly rising liver function tests, but normal HbA1cs, normal glucose, slightly elevated blood pressures, and not really knowing what to do with them. And I think the beauty of what you've covered today is we now have a clear message on what to do. And I think with the testing, we can pick up metabolic syndrome maybe 10 years earlier than we previously could.
Definitely.
So the effect of cost saving for the NHS could be extraordinary.
Information is king, so we have to measure to generate the useful information. And if you generate a lot of information, these measurements could even be further refined, maybe even cheaper later based on big data.
Okay, this is brilliant. So have you any, if you were to summarize what we've talked about, what are the key take-home messages for, let's say, either a physician or a patient out there who says, I want to improve the metabolic health of my patients?
Yeah, I think two major points. The first is do postprandial measurements after meal or at least glucose. The second one, try to incorporate lipid measurements into the practice, because lipids often derange a lot earlier than glucose. So if even the fasting metrics can be improved by incorporating triglycerides. There is, for example, this McAuley index. It uses the triglycerides together with the fasting metrics, and then there is the triglyceride glucose index, which is also a very cheap and accessible metric to determine insulin sensitivity, insulin resistance, and these are not really used. These are available, more accessible than, for example, insulin. We can also do now cheap insulin testing within the mentioned project of metabolic, but it's a bit more cumbersome. You need more blood. It requires a little bit more expensive test. It's already accessible, but almost the same, not the same, but a similar level of quality of information can be obtained by including the lipids into the metrics and indices.
And is the triglyceride glucose index as simple as putting triglyceride over glucose, and that gives you
there is a simple equation or formula. There is a simple formula. Then you just need to substitute in your measured fasting glucose and fasting triglycerides.
Okay,
That's it.
So if we Google it, we'll find it.
Yeah, yes. Feel free to contact me, but it's a 5-second search indeed.
Superb. And for the patient, so if you were to give a simple message to a patient listening to this to say, you know, it appears my triglycerides are higher than they should be. My sugars are okay now, but actually I've got 3 people in my family who have type 2 diabetes. What simple things should I do today to improve my metabolic health?
Yeah, pressure your GP. Most people don't have a good understanding of the underlying mechanism, the metabolism, physiology, and these kind of things. But I think everybody can understand that knowing more can be profoundly useful. So having more information, more data, and using more of these indices, like the HOMA for insulin resistance, the triglyceride glucose, the McAuley, and this kind of relatively simple can substantially improve the quality of information we see. And we can close the huge gap between measuring fasting glucose, for example, and doing a research type of hyperinsulinemic euglycemic clamp, which is several hours of infusing different things and measuring all the time different things. So one is the gold standard, of course, in a research setting. Yeah. But otherwise, we should be really working on to get out the most useful information with the least possible cost and efforts. That's kind of a Pareto principle in engineering, but I don't see any reason why it couldn't be applied to medicine.
Yeah, and I think the message I was looking for patients really is to eat food in the most unprocessed way possible. So keep the structure of the original food as it was in nature, and then if you eat that sort of food, you're less likely to do harm to yourself.
There are a few rules. It can be dumbed down into a few rules. I would leave out the most offending types of foods or food components. For example, if you can leave out wheat, barley, and rice, these are the most strongly processed and also the most quickly absorbed, digested types of carbohydrates in our food chain. If you can leave out these 3, and then you can limit simple sugar consumption. And what you still include should go to the end of the meal as dessert. Never start a meal with white bread or a sugary stuff. Do not snack, because that's basically starting a meal with highly processed foods. So yeah, have a good sleep. That's also very important. Try to finish your daily meal well before going to bed, so that you don't go to bed. Don't stress your sleep quality. Reduce your sleep quality. These are a few very basic. So food order, meal frequency, leave out the worst perpetrators, and good sleep, and yeah, do some exercise, even just stand up every hour for 5 minutes and stretch out, walk around a little bit if you are in an office. These are important and very useful interventions. And it's not this regimen, if you put it together, it's not a drastic one. It's relatively easy to follow. At least I believe.
Yeah, they're fantastic, simple messages any patient and in fact any clinician can take home just to improve the health population that they're serving.
Yes.
So I think that's been a absolutely fascinating and brilliant talk, and I'd like to thank you very much for your time. So we're going to keep this recording and we will put it out on some social media outlets. And anyone who's listening to this, they please get in touch with me or Gabor, and we can have further conversations, particularly around the testing and the triglyceride glucose index that can be got from this simple machine on a finger prick test.
Yes,
Brilliant.
knowing more is very important.
Brilliant. Thank you.